Hi, everybody. This is Dr. Kerry Kriger. I'm the founder and executive director of Save the Frogs, and I'm very happy to be here today to talk to you about atrazine, which is one of the world's most commonly used pesticides. And this is a class being held by Save the Frogs Academy. For any of you who have not attended one of our online classes, these classes are taking place every Wednesday at 11 a.m. San Francisco time and every Sunday at 5 p.m. San Francisco time. And we talk about a whole range of topics related to amphibian conservation. So today we are focused on atrazine, which is one of the pesticides that has had the most amount of scientific research done on it. It's been banned in the European Union since 2004. It's been out of use in Italy, Germany, and France for much longer than that. And it has been in use for at least 50 years. I guess before I continue, give me 1 second. Okay, so I just wanted to introduce myself and so that you can see who I am. These are strange classes as they're online. The great thing about online classes is we can reach people all over the world. So what we want to do is not just teach you about atrazine, but get you involved in helping us get atrazine banned, federally banned and out of production at a minimum in the USA. But as I said, atrazine's in use in about 50 plus countries around the world, contaminating our water where amphibians live. So this is a picture of atrazine. I found this atrazine in Ghana. This is Save the Frogs Ghana executive director Gilbert Adam. There were a lot of pesticides being sold in Ghana all over the streets. There were even billboards up promoting pesticides. So it's definitely not just an American problem. One of the benefits of getting atrazine banned in the USA is that a lot of countries around the world look towards the USA for direction on what to do with regards to certain pesticides. So a ban of atrazine in the United States would certainly have a ripple effect and cause some other countries to ban atrazine as well. So today we've got a very special guest, Dr. Tyrone Hayes, on the line, and he's been doing atrazine research for a very long time. He's currently at UC Berkeley, and he's going to be talking to you about atrazine. Before we get him on, I just want to give you a brief history of Save the Frogs and what we have been doing to raise awareness of atrazine and help get it banned. I personally first heard about atrazine when I started my PhD research in 2003. I was in Australia and heard some pretty bad things about atrazine. And in 2004, I believe Dr. Hayes published a very well-known paper about the effects of atrazine on amphibians. And so I've had an interest in in atrazine ever since then. And so I wanted to get Save the Frogs involved in getting atrazine banned. And I think atrazine is the most important pesticide for us to focus on, not just because it has such harmful and scary effects on amphibians, but because it's so commonly used. It's the most commonly detected contaminant or pesticide in U.S. groundwater, tap water, and rainwater. And because it's on our number one crop, our most used crop, our most important crop in America is corn, and most Americans eat corn most corn that is not organic is probably sprayed with pesticides containing atrazine. So it affects so many Americans. It has such scary effects on amphibians that we'll hear about shortly. So I really think it's a fantastic way to educate Americans about the harm that can be caused by pesticides, because really atrazine is just one of over 18,000 legal pesticides in America, and a lot of those are exceptionally bad for amphibians. So just raising awareness about pesticides and getting people to be smarter consumers, to eat more organic food, to buy cotton that's not covered in pesticides. This is a great way to clean up America's water and ecosystems. So the atrazine campaign is a great way to just get people thinking about pesticides in general. So on the third annual Save the Frogs Day, which was April 29th, 2011, we held a rally in Washington, D.C. that was organized by Brianna Binder, who's now our Pacific Northwest coordinator. And I wanted to be in D.C. because the U.S. Environmental Protection Agency is based in Washington, D.C., and I want to make sure that politicians know about atrazine because they're the ones who can quickly get it banned. The administrator of the US EPA could sign a piece of paper and atrazine would be banned. The president of the United States could sign a piece of paper and atrazine would be banned. And even aside from those things happening, just by going through normal EPA measures, atrazine can get banned. And this class right now is taking place specifically because we've got 10 days to submit comments to the US EPA for an official call for comments period so that it can go through their channels and EPA could get banned or technically not re-registered. Every 15 years in America, a pesticide must get re-registered by the EPA. So if they approve a pesticide, every 15 years they have to review current science on it, make sure that it's still not harmful, and then they would register it or not register it. So right now the EPA, or atrazine, is in its 15th year. So the EPA has to review atrazine. So we've got 10 days for this call to comments, and I'll talk about that shortly and how you can take part. And so this is at the start of our rally. We started the morning by getting greeted by the officers from the Department of Homeland Security. This is me with Dr. Tyrone Hayes. And I thought this was very appropriate because we were all there for Homeland Security. We, Save the Frogs, me and you, are defending our homeland from harmful chemicals and protecting the wildlife and ecosystems in the water that we drink and the food that we eat. And we got about 40 people there for the Save the Frogs Day rally. It was right at the steps of the US EPA in downtown Washington, D.C. And we also got a lot of news journalists there, so made a lot of news. This picture proves that kids can help save the frogs too. We had a volunteer from New York come, so we had people from a lot of states showing up. And Dr. Tyrone Hayes was our special guest presenter there and gave a great talk on atrazine. And then we all took a march through DC to raise some awareness and get people thinking about frogs and atrazine. We had a police escort in the street helping us, helping stop traffic for us. And then we went to the White House to get President Obama involved. He didn't come out, but the EPA scientists heard about the rally and invited me to come speak a week later to their Office of Pesticide Programs, where I gave a 50-minute presentation on atrazine and why it should be banned. And I delivered over 10,000 petition signatures to the EPA. We still have this petition. You can sign it electronically on savethefrogs.com/atrazine, which we'll take a look at in a couple minutes. And also, you can download a PDF of this flyer and print it and collect signatures and send it in to us. So we're still collecting signatures to get atrazine federally banned. And based on that petition that I delivered, the EPA held a call for comments in 2011, in I think November of 2011. And we got a lot of Save the Frog supporters writing them. In 2012, the EPA had a scientific advisory panel hearing on the effects of atrazine on aquatic health and aquatic wildlife. And I spoke there. I gave a 25-minute presentation, and I was the only person who spoke there who was speaking against atrazine. There were about 25 people from the atrazine and the farm lobby, people who make a lot of money by selling atrazine, and they spoke. 25 people for atrazine, and I was the only person against atrazine. And that kind of shocked me, scared me a little bit, because we need people who are willing to, at a minimum, go to these events where we're invited to submit comment. The public is invited, and people need to get out and about and do things if we expect to get pesticides banned, because the opposition, Syngenta, the company that makes it, who's based in Switzerland, where atrazine is actually banned, they've got $11 billion at stake. That's how much they make each year. So they make sure they're at these kind of events. And if all the EPA politicians ever hear is pro pesticide lobby, then they're just going to keep pesticides legal. So we need to be active and heard. I know that most Americans who know about atrazine want atrazine banned. So we need to make our voices heard. And I strongly encourage you all to go to our atrazine webpage, savethefrogs.com/atrazine, and download an MP3 of my presentation. Or you can go to our YouTube channel, youtube.com/savethefrogs, to listen to my presentation. So right now, as I said, the US EPA is seeking your input. There's a call for comments that goes through August 26th. And that's your chance to tell the EPA what you think. So what I'd really love for every person on this call to do is afterwards, when you know so much about atrazine, because Dr. Tyrone Hayes is about to teach you firsthand all about atrazine, go to savethefrogs.com homepage, and there's a link right at the very top so that you can click straight through to the government webpage where you can submit your comment. If you go to our atrazine page, savethefrogs.com/atrazine, you're welcome to grab some text from there, get some ideas, just put a paragraph or two together for the EPA, make it personal, because if they get 10,000 of the same exact comment, they really only think about it one time. So make it personal. They will read it. Okay, and we want volunteers helping us, not just by submitting their comment before August 26th, but we need some volunteers working on this campaign. Save the Frogs only has a few total staff members. We're really dependent on volunteers. It would be great to have a campaign leader who can take charge of the atrazine campaign with supervision from me and commit to at least 1 year. We also need some assistants. So if you want to help out and have your Save the Frogs volunteer focus for a year be atrazine, if you've got about 4 hours per week to dedicate, then start thinking about what you'd want to do, and let's talk. And before I go on, I just want to put out a big thanks to Save the Frogs volunteer graphic designer Ellison, who came to our rally in DC and pretty much any of the cool graphics you see on our site and any of these Save the Frogs graphics, she probably helped design in her free time. And she's a huge asset to Save the Frogs. It's my goal to have enough funds to hire her full time because our programs, our marketing, all of our campaigns depend on having really great graphic design that's eye catching, that people will spread around on their websites. And it's a huge benefit. So it would be really great if all of you can donate to Save the Frogs to help us get all of our campaigns going by having really amazing graphic design, not just as a part-time volunteer thing, but as full-time happening. I know that all of our international, our chapters internationally all need graphic design assistance. I need graphic design all the time, so please help out. Thank you so much. And so I'll come back to all this to update you about upcoming events. Right now, I'm going to switch it over to Dr. Tyrone Hayes. So give me one moment to get this technology going on, and I will let Dr. Hayes introduce himself and talk to you about atrazine. Give me one moment. Thanks. Hi, Dr. Hayes. I have unmuted you, and I'm switching to your screen.
Okay, can you hear me? Can you hear me? Can you hear me? Can you see the screen? Can you hear me?
I can hear you.
Okay, can you see the screen?
I sure can.
Okay, should I go ahead and get started?
Yes.
Okay, well, welcome. I want to thank everybody for joining us. What I'd like to do today is to tell you a little bit about the impact, the effects of atrazine on amphibians, the impact of atrazine's role in amphibian decline, as well as what atrazine's effects on amphibians are telling us about the impact of chemicals on wildlife, especially atrazine on wildlife in general, and also the role that atrazine might be playing in declining health in humans even. I also first want to acknowledge the funding. As a matter of disclosure, I want to point out that I have been paid previously by the manufacturers of atrazine. So that's a matter of my disclosure, and just so that everyone knows that I initially began studying atrazine because the manufacturer of atrazine, Novartis, and then Syngenta, actually funded me to work on atrazine. Atrazine is shown here. If you can see the slide, it's a still called S-chlorotriazine. It's the class of chemicals it belongs to. Atrazine is an herbicide, mostly used on monocot crops. In this country, the biggest use of atrazine is for use on corn, as you heard earlier. Atrazine has been used for, I should update the slide now, I guess, for 50 years now. We use 80 million pounds annually in the US of atrazine. It's used in more than 80 countries, but it's now outlawed in all of Europe. Again, as you just heard, the European Union has declined to re-register atrazine, which is an interesting observation because the manufacturer of atrazine, Novartis and now Syngenta, its home is in Switzerland. I again began working on atrazine in 1998, and that's when I was asked by the manufacturer to study the effects of atrazine on a laboratory amphibian model, in this case the African clawed frog, or Xenopus laevis. Initially, we showed with funding from the manufacturer that atrazine inhibited growth of the voice box or the larynx in males. which is significant because the same reason that men have deeper voices than women, because of testosterone that causes our larynx or our voice box to grow in us humans. Testosterone plays the same role in frogs. It causes the voice box to grow, such that male frogs sing and female frogs don't. To try to understand why animals exposed to atrazine might be limited in the amount of atrazine they produced, we examined the gonads or the sex organs of these frogs, and we showed the following. So here are the kidneys of an animal, of a frog that's been exposed to atrazine, that have been dissected. And on top of the kidneys, you can see the testes. You can see that this animal also has ovaries and more testes, as well as another set of ovaries. So what we found was that atrazine not only demasculinized or took away the male typical characters of frogs, but it also feminizes frogs by causing genetically male frogs to develop ovaries in addition to their testes. We then proposed, after this first study, that the reason that atrazine both demasculinized and feminized frogs was the following. Normally the testes, as symbolized by this yellow oval in this slide, makes testosterone. In fact, testosterone, a so-called male hormone, this word literally means testicular hormone. It's two words pressed together. Atrazine induces the enzyme or the machinery, if you will, called aromatase, that converts testosterone into estrogen, the so-called female hormone. Our proposal was then that the amount of testosterone in an exposed male is reduced because the testosterone is being converted to estrogen. Therefore, they're demasculinized, but they're also subsequently feminized because now males are making the female hormone instead of the male hormone. We showed in that same study that we published almost 11 years ago now that indeed atrazine-treated males had reduced testosterone levels. So you can see the really low testosterone levels in males that have been exposed to atrazine, and those testosterone levels are different from a female. So they've been demasculinized to the point that their testosterone levels, their male hormone levels, are no higher than what you would observe in a female. We published that original study in one of the most prestigious journals in the country, in fact, the world, the Proceedings of the National Academy of Sciences, and that study published was entitled Hermaphroditic Demasculinized Frogs After Exposure to the Herbicide Atrazine at Low Ecologically Relevant Doses. A couple questions weren't answered in that study. One is we didn't know if the hermaphrodites were males with ovaries or females with testes, so we couldn't be sure which species or which sex of the species had been affected. We also didn't know what are the consequences. So what happens when these animals become adults? We didn't follow the animals all the way to adulthood in this first study. In a subsequent study we did, we showed that animals that are exposed to atrazine with the reduced testosterone have a reduced ability to attract females, to attract mates. So their reproductive success was severely impaired. We also showed that of those animals, when they were in a competitive situation, the atrazine-treated animals on average had much lower testosterone levels. And what you're looking at now, the individual testosterone levels for males, and the hearts here show the males that successfully copulated with females. So what you can observe here is that the atrazine-treated males almost never were successful in attracting females. The two hearts you see on the red on the atrazine side are the only two males that were ever able to successfully attract females. In other words, atrazine maintained testosterone levels so low in those exposed males that they weren't able to display male reproductive behavior. They were unsuccessful at mating. Again, these are the data showing that atrazine-treated males are almost never successful in attracting females. What's more, if males are individually housed with females, we showed that the atrazine-treated males were incapable of fertilizing eggs, so their fertility was severely impaired. So, for example, here are control males showing that a control male or unexposed male is able to fertilize about 85% of the eggs when it mates with a female, whereas an atrazine-treated male has a fertility rate that's about 15%. If you examine the testes in cross-section under the microscope, what you find is that the control males have sperm in the testicular tubules, so the arrow here are showing the sperm inside the tubules, whereas the atrazine-treated males typically have testes that are void of sperm. There's not enough testosterone to produce sperm. In a below section up here, magnified, you can see now the dark arrowheads. These are the sperm inside the testicular tubules, and as compared to a similar section in atrazine-treated males, where there's only cellular debris in the testicular tubules. So even if the males displayed mating behavior, even if they tried to breed, tried to mate with the females, their sperm count is so low that they typically have a very low fertility. Furthermore, what we showed is that we identified a gene, that's what's demonstrated here, that's only present in females. So we could determine who's a genetic male and who's a genetic female. The females express aromatase in the controls, which again, the aromatase is the enzyme, the machinery that allows the female to produce estrogen. If you examine the atrazine-treated animals, what we're showing is that some of the females, or the atrazine-treated animals, some of the genetic males, which are missing this gene, completely turn into females morphologically. They express aromatase and they make estrogen as if they are female. So a proportion of the animals are completely feminized. They completely develop into females as shown here. So this is a normal male, a control male. This animal is actually a genetic male that's actually breeding, that's copulating with this male. Even though it's a genetic male, it actually functions as a female and even lays eggs. So atrazine has the capacity to not only demasculinize or to take away male characteristics, to take away male behavior, to take away sperm from the males, but atrazine also has the capacity to completely feminize genetic males and make them turn into females. Again, the second paper was published in Pursuits of the National Academy of Sciences, again, one of the top 3 science journals in the world. And the title of that paper is Atrazine Induces Complete Feminization and Chemical Castration in Male African Cloud Frogs. In more recent studies that we are just in the process of publishing now, we showed that atrazine not only demasculizes males, but it has the capacity to induce males to behave like females, even when they have testes. So, for example, what you're seeing here is a genetic male that still has testes, but that has chosen to copulate, to mate with another male. And we have a number of examples like this in the laboratory. Almost never occurs in unexposed animals. Furthermore, what we've shown is that the males who are copulating with other males aren't simply small males that are being overpowered. The males that behave as females, are shown here, are often larger than the males that they copulate with, so they're not being forced, if you will. What we've also shown is that the males that behave as females typically have lower testosterone than the males that they're copulating with. But furthermore, we've shown that the males that are behaving as females has estrogen or female hormones that are more similar to a female than to a male. We've also shown that some of those males have partial feminization in their morphology and their body parts. So here, for example, are testes from an atrazine-exposed male, and what you're looking at here, if you follow the arrow, is these are oviducts. So this is the equivalent of a man with a uterus. So they are genetically male, they have testes like a male, but they have other parts that are more similar to a female, including their behavior, for that matter. In another paper that we published in the scientific journal Nature, we showed that atrazine at very low doses induces gonadal abnormalities. It causes undeveloped testes as well as testicular eggs. So in the North American leopard frog that we studied, males exposed to atrazine maintain their testes, but they developed eggs in their testes. And an example of that is shown here. So here are testes from a dissected male, and all of these lobules that you see in the testes are eggs that have developed in this male's testes. And in fact, those eggs are being yolked up and are bursting through the surface of this particular male's testes. To give you an idea of what levels we're talking about, atrazine is effective in the lab at 0.1 parts per billion. That's the equivalent of about 1/1000th of a grain of salt in 2 liters of water. To give you an idea of levels of atrazine that are found out in the wild, atrazine is applied, the recommended application is 2.9 to 29 million parts per billion. So in other words, atrazine is applied at levels that are 290 million times its biologically effective dose. What you're looking at now are minimum and maximum detected levels of atrazine in agricultural runoff, temporary pools, permanent water, and precipitation. So atrazine can be present in rainwater at levels that are higher than we know can produce these types of effects that can cause hermaphroditic development in frogs. So in other words, all of these types of environments, from agricultural runoff to rainwater, as shown in the red overlay, can have enough atrazine to produce adverse effects on amphibians, and what EPA considered safe, 300 parts per billion, and what the EPA considers safe in drinking water, 3 parts per billion, is 30 times higher than what we know to be biologically effective in frogs. In another study also published in Nature, we showed that these types of effects occur in the wild. So this isn't just a laboratory artifact. So for example, what you're looking at here are kidneys, and these are the testes from a frog. And if I show you what those testes look like under the microscope, what you see are that eggs have developed in the testes of this frog. And this is a frog from the wild, not a frog from the laboratory. So I'm using the arrow here to show you eggs instead of sperm that are developed in the testes of a frog exposed to atrazine in the wild. We conducted a study where we examined leopard frogs across the United States. And in the red, we showed that everywhere we found hermaphroditic leopard frogs, we found atrazine and vice versa. This study, which was published in Nature, was combined with a laboratory study that showed that, for example, we can take animals from a site where they're hermaphrodites and raise them in the clean in the laboratory, and they would develop normally, and that we could also take animals from a clean site and raise them in a laboratory exposed to atrazine and show that we get hermaphrodites. So in other words, this was not just natural variation. So I've often called this talk, as you saw from the beginning, from silent spring to silent night. And I call it that because in much the same way that Ray Carson in Silent Spring thought that the death of birds due to pesticides, in that case DDT, was a warning. Our pending Silent Spring was a warning to us. In much the same way, I think that our pending Silent Night, or the death of frogs, is also a warning to us. For those of you who don't, I'm assuming most of you on the call know that as much as 70% of the world's amphibians are in decline in what is many scientists consider the 6th mass extinction on Earth. The link to humans, I believe, is manifested here most clearly. This is a slide from Lake Nabugabo in Uganda, where the runoff from this crop goes into these containers and is the sole source of drinking and drinking water and cooking water, bathing water for a nearby village. What I think this slide illustrates is the link between environmental health and public health. So, for example, if the frogs and fish that are living in this water are unhealthy because of the chemical that's being used in this crop, then that impact would directly impact the humans that are using water. The problem is, I think, in the United States, is that we don't necessarily relate to our environment in the same way. So, for example, in Oakland, where I live, my water comes from here, as the arrow's pointing, but because I don't collect that water myself, that connection between environmental health and public health is not always clear to us. That connection has been revealed by much of the science on atrazine. So, for example, a colleague of mine wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence of causation. What I'd like to show you briefly now is that not only have we shown that the effects of atrazine occur in more than one population, but occurs in multiple species of frogs, as I've already told you, multiple families, and in every class of vertebrates that's been examined. So atrazine has similar effects not only in amphibians, but in fish, reptiles, birds, and even analogous effects in mammals, including humans. that I'll briefly tell you about here. In part, that information comes from a paper that I published with multiple authors. I believe there were 22 of us from multiple countries, from at least 12 different countries around the world, showing that atrazine has adverse effects as an endocrine disruptor in every vertebrate class. To give you an idea of some of the things we showed not only does atrazine reduce testosterone and sperm production in frogs, but that effect occurs across vertebrate classes. So, for example, I showed you that atrazine caused a decline in sperm in amphibians, but similar data from Belgium show that atrazine causes a decline in sperm in fish. Atrazine causes a decline in sperm in reptiles. This is data from a caiman, which is like an alligator from a research in Argentina. Atrazine causes a decline in sperm in rats. So, for example, on your left, this is a testicular tubule from a rat filled with sperm. On the right is a testicular tubule from an animal that's been exposed to atrazine that's devoid of sperm. And those data have been repeated in both Croatia as well as Nigeria. And a colleague from Pakistan has showed that similar effects occur in birds. So this is the section of testis under the microscope in a quail. And on your right is a testis from a quail that's been exposed to atrazine. So this decline in sperm occurs across vertebrate classes, across multiple species. In humans, a colleague showed that there's a significant relationship between low sperm count and atrazine level in humans. Now, again, this is just a correlation, except that experimental evidence in everything from fish to birds to amphibians to reptiles to laboratory rodents shows that atrazine causes a decline in sperm count. And here now we have a correlation showing a decline in sperm count and a decline in fertility in humans. What's more is, if you look at field workers, atrazine levels are even higher. And if you look at men who apply atrazine, levels of atrazine in the urine of men who apply atrazine are 24,000 times higher than we know to be associated with poor reproductive health in men, 24,000 times higher than what we know to be biologically effective in laboratory animals. This raises concern in particular because most of the people who work in agriculture are Mexican Americans in this country. So there is an issue of environmental racism or environmental justice issues. So, for example, in California, we are the 5th largest economy in the world. 1 in 10 jobs in agriculture, 30% of the land is in agriculture. We produce 350 agricultural products. Half of the U.S.'s food is produced in California. We use more pesticides than any other state, and 90% of the workers, though, are Hispanic. As a result, if you look at the top 10 counties for agriculture in California, the 30% that makes us the 5th largest economy in the world, most of the top 30 poorest towns in California fall within those agricultural counties. So in other words, the people who make us the 5th largest economy in the world are the unintended targets of atrazine and its effects. On the other side of the equation, atrazine not only induces aromatase and estrogen production in frogs, which leads to phylogenesis, or egg yolking, and oogenesis, or egg production, but aromatase and estrogen are also associated with breast cancer and prostate cancer. With regards to prostate cancer, there's an 8.4-fold increase in prostate cancer in men who work in Syngenta's factory making atrazine. And this is a community that's 80% African American. There's also a significant correlation in at least one study between women whose well water is contaminated with atrazine and breast cancer. Women who don't drink that well water but live in the same communities don't have the same incidence of breast cancer as women who live in those communities and drink that well water. Again, that's just a coincidence, but if you look at rats, atrazine causes a decline in testosterone in exposed rats, and atrazine causes an increase in estrogen in exposed rats, and breast cancer is an estrogen-dependent cancer. Furthermore, in rats that are fed atrazine, there's a significant increase in the breast cancer, the mammary cancer, that those rats experience. So atrazine is associated with an increase in breast cancer in humans. And a study all the way back in 1994 showed that atrazine contamination in drinking water causes rats to develop more breast cancer. What's more is if you take human cell lines and expose them to atrazine, the atrazine induces aromatase activity and estrogen production, just like it does in laboratory fish, frogs, reptiles, and mammals, rats. The company that produces atrazine, the factory that produces atrazine, is located in San Gabriel, Louisiana, in the United States. And there's a pipe, and again, this is a community that's 80% African American. There's a pipe that empties directly into the Mississippi River, and it turns out 1.2 million pounds of atrazine flow into the Gulf of Mexico every year down the Mississippi River. This raises concern because 11 out of the 13, if you look at the top 13 cancers in the United States, African Americans and minorities in general are more likely to get 11 out of the 13 top cancers. What's more is African Americans are more likely to die from 100% of the top cancers in the United States. This raises a question of whether or not there's a genetic difference, there's a biological difference, or is it because minorities are more likely to live in and more likely to work in areas where they're exposed to chemicals like atrazine that we know are associated with these adverse outcomes? In other words, is the information from frogs, which develop in an aquatic environment, telling us something about humans, which also develop initially in an aquatic environment and depend on the same hormones that atrazine causes problems with? In a related study, we've shown that atrazine reduces aromatase in breast cancer cells, and that's significant because aromatase and estrogen production locally in breast cancer cells is known to promote the spread and development of tumors in women who develop breast cancer. In fact, the #1 treatment for breast cancer right now is a chemical that actually knocks down or eliminates aromatase, thereby reducing estrogen so that breast cancer damaged cells don't spread. This, of course, presents a problem because atrazine does exactly the opposite. It induces aromatase, and it increases estrogen, and it's associated with the promotion and the increase of breast cancer, both rats and in humans. Turns out that the same company, Novartis, produces both chemicals. As you can see here, their advertisement shows that they offer treatments for cancers that range from breast cancer. So the same company that produces an aromatase inducer, atrazine, that's associated with breast cancer, also produces an aromatase blocker, letrozole, that's used to treat breast cancer. Such that if you're living in the Midwest taking letrozole to treat your breast cancer, how is that impacted by a chemical produced by the same company that induces aromatase, increases estrogen, and that's associated with breast cancer? The manufacturer has taken a number of steps to try to prevent this information from becoming public and also try to discredit me. So, for example, they've hired PR firms to attack my data, to attack documentaries that I've been involved in. Here's another piece from the media where Syngenta investigated the press and shaped the news about atrazine. And what I'm showing you now are documents that were recently uncovered in a court case against Syngenta in their own handwriting, showing that their main goal in doing research was to, as you see here, discredit Hayes. Not to fully study and examine atrazine and produce the truth, but rather to discredit my work on atrazine. One of the other problems is that the seed industry is pretty much owned by the pesticide industry. So 90% of the seeds, as you see this figure shown in blue, are owned by pesticide or chemical companies, as you can see in red. So, for example, there's Syngenta shown here. Another part of the problem is that the government workers that are actually supposed to be regulating pesticides are actually also some of the same individuals that are involved in producing the pesticides. Sorry, there seems to be a hang up with the PowerPoint here. Sorry about that, folks.
Tyrone, if it's easier, you're welcome just to continue speaking without the PowerPoint.
So the last things that I was going to get at are, sorry about the technical difficulty. I think I was trying to log on to something. The last part I was going to speak about is that more recently there have been a number of studies linking atrazine to birth defects in humans. So not only are we talking about, and we're back online now. So not only are we talking about problems with wildlife, as I mentioned earlier, but there are reproductive effects in humans as well as, and here we're back online. Here, as well as a number of birth defects associated with atrazine exposure. So there's studies showing that atrazine causes prostate and mammary cancer in rats, which are a model for us. Atrazine causes immune failure in rats. Atrazine causes neurological damage when pregnant mothers are exposed. And most disturbing, atrazine causes abortion in rats because of the hormonal problems associated with atrazine exposure. Of those rats that don't abort, the sons are born with prostate disease. The daughters of exposed rats are born with impaired mammary or breast development, such that the second generation shows up with retarded growth and development. In other words, this rodent, and this was a study done by the Environmental Protection Agency, suffers from atrazine that its grandmother was exposed to, even though this rodent itself wasn't exposed. The link to humans has already been made. So, for example, colleague Paul Winchester has shown that birth defects peak when women are become pregnant during peak atrazine contamination periods. Some of those birth defects include, and I'm reading here now from a paper published by the Center for Disease Control, include maternal exposure to surface water atrazine associated with fetal gastroesthesis. And this is what that disease looks like in humans, what that birth defect looks like. And another more recent study showed, and again, this is from the National Institutes of Health, that maternal exposure to common herbicide atrazine is associated with a nearly twofold higher risk of having offspring with a rare birth defect of the nasal cavity. And this is what that birth defect looks like. So again, there are multiple birth defects, in particular two almost deadly birth defects associated with exposure to atrazine. The manufacturers responded that they assume no obligation to update forward-looking statements to reflect actual results. And most relevant to this conversation regarding my work, the EPA has stated that the ultimate decision, whether or not to ban atrazine, is much bigger than science. It weighs in public opinion. So for those of you listening today, registering during the time that you can comment now with the EPA is incredibly important for getting this dangerous chemical off the market. I maintain a website that, or I should say maintain, I have a website that I don't maintain very well, but you can get information from there. There's also a Facebook page where a great deal of atrazine information is disseminated, and there are a number of petitions, one that you just heard about, that you can sign on to. And a bill to ban atrazine has been reintroduced by Congressman Keith Ellison, and you can write to Congressman Keith Ellison and assist in getting atrazine off the market by supporting the bill that he's just presented to Congress. So thank you very much for your time, and I hope that together we can make the world a much cleaner and a much safer place for ourselves, for our children, and of course for the frogs. Thank you.
Thanks a lot, Dr. Hayes. Give me 1 second to switch the screen back, and then we'll take questions from people. So if you have a question and you want to send it in via your chat, you can do that. If you have a question and you have a microphone, then you can raise your hand and I can potentially unmute you. But let me first deal with this technology and get my screen back. Okay. Kathleen Franco, I see your hand up. So I'm going to unmute you. And if you have a question, you can ask me or Dr. Hayes. Kathleen, you are on the line. How are you doing?
Hi, I'm good. I have two questions. My first question is whether the males that convert to females, if they can produce viable offspring.
The answer is, can you hear me?
Yes.
Yeah, I can hear you.
Yeah, the answer is yes. So those males that completely convert to females, they can produce viable offspring. But in this species of frog, the males only have one type of chromosome. So those males can only produce male offspring. So only males can develop from the eggs that they produce.
Okay, I see. And my second question is, if you know of any chance what the half-life of atrazine is in our current drinking water.
Oh, that's a good question. So all I can tell you is that there are a great deal of numbers floating around. But in Europe, where like France, where atrazine has been banned for over a decade, the levels in the drinking water in the aquifers haven't changed in those 10 years. So atrazine can be around, depending on the, whether it's in surface water or groundwater, can be around for over a decade, maybe 2 decades.
Wow, okay, great. Thank you very much.
Okay, so we have some more questions coming in. We have one, Kaser Alarcon. Kaser, sorry if I got your name pronounced wrong. You're unmuted. Do you have a question?
Hello.
Hi, do you have a question?
Oh, this is Tyrone.
Oh, sorry about that. Okay, so it looks like no more people want to ask a question on their mic, but I have some questions that have come in from the chats here. So I guess we'll start. Let's see. People have been asking about slideshows or these getting access to these slides. So I know that my slideshow will go up on savethefrogs.com/slideshows, and we'll add a link to savethefrogs.com/atrazine. And I'm not sure what Dr. Hayes's plans are with his slides, but I'll talk to him later. And if he is able to give us those slides, I can add them to the atrazine page as well, so that if people out there want to take another look at them, then they can do that. So just stay tuned. And we'll also send you out some links, everybody who's registered for this webinar. So let's, I have a question for you, Dr. Hayes. What do you think the number one thing that people out there listening can do, and or that Save the Frogs can do to get a atrazine ban to make it a reality?
Well, first let me comment on the slides. There's a number of, I can send you links. There's a number of videos of very similar lectures that I've given online as well, and I'm more than happy to provide the slides. In terms of doing something for atrazine, I think writing in this comment period, and if you're able to, even showing up at the EPA hearings and presenting your opinion on atrazine would be very helpful. Writing into Keith Ellison in support of the bill to ban atrazine is also very helpful. And of course, signing the Save the Frogs petition and having your name, having your opinion added to ours and delivered to the EPA is also very important.
Okay, thank you. Let me keep looking through some of these questions. Okay, you probably can answer this better than I can, Dr. Hayes. Someone asked, has anyone sued or attempted to sue Syngenta? And I know there's been some lawsuits in the Midwestern USA. Do you want to say anything about the history of lawsuits against Syngenta?
Yeah, there's been one major lawsuit. The Corey and Tillery, the law firm, sued and Syngenta settled for $105 million. There's another lawsuit pending from Corey and Tillery. There's also a lawsuit in Hawaii that involves atrazine contamination of an entire town and a school. So those things are happening as well.
Okay. On that note, I want to add, I don't think of Hawaii as being a place with corn, but I do know that there's a lot of sugar and sugar cane that's grown in Hawaii. So I'm assuming that's what atrazine was used for there. When I'm talking about atrazine and when people talk about atrazine, they think about corn because that is the number one crop it's used on, but it's also used on sugar cane, rice, millet, Christmas trees, golf courses and in lawn care. So it's in a lot of places. And so since Hawaii got brought up and that's likely sugar cane, I'll just say to everyone out there, one incredibly easy way to help save the frogs is to reduce your intake of sugar, because sugar cane is often grown where rainforests used to exist, but it got chopped down to grow that sugar cane. And that's throughout much of the tropical world where there's very high amphibian biodiversity. Rainforest gets chopped down, they grow sugar cane, and they spray it with bad pesticides.
So Kerry, what's interesting is it used to be used in Hawaii for sugarcane, but sugarcane is not the major use in Hawaii now. It turns out that Hawaii has sort of lax rules, and they can grow year round. So the corn industry has actually has bought up a lot of land in Hawaii, and they're actually growing their experimental GMO crops in Hawaii and using pesticides. Hawaii was the only state that did not abide by the Clean Water Act. So many of the companies, Syngenta and Monsanto, have moved into Hawaii and are growing corn here, the GMO crops, to produce the seed, and then that seed is shipped around the world. So it's a really interesting abuse of land and of a way to get away from laws that we abide by in the mainland states.
Okay, thanks for that clarification. On that topic, and because I know a lot of people are interested in GMOs, they're in the news a lot, and they're, to me, pretty scary, changing the DNA of the food that we've evolved to eat. Is there any relationship between atrazine and GMOs? Are they making crops that you can really douse with atrazine and genetically modifying them for that purpose?
I don't. I mean, the biggest GMO pesticide combination, of course, is Monsanto's Roundup, and the value of atrazine, and the reason that it was so heavily used previously, is that corn and sugarcane and grasses are naturally resistant to atrazine, so it didn't require genetic modification. And the reason that Roundup has taken off and now become the #1 herbicide is because now more and more crops are being modified to be resistant to Roundup.
Okay, thanks for that. So I think we have one final question for you, if you have a second. Do you have time for one? Okay, so this is from Spencer Siddons, who used to head our University of Wisconsin Stevens Point chapter of Save the Frogs. And Spencer, you are on the line. How's it going, Spencer?
Hello, it's going well. Thank
Hi.
you. Can you hear me?
Yes.
All right, great. Yeah, my question, I guess, is kind of for both of you, whoever can answer it. I was wondering if either of you are aware of any interactions between atrazine and the citric fungus, BD, and maybe if it inhibits growth or of BD on amphibian skin or anything like that.
Okay.
That's interesting.
Go on, Dr. Hayes.
I was going to say there are a number of studies, including some of my own, that show that pesticides, especially mixtures of pesticides, lower immune function, probably by increasing stress hormone response, and thereby increases an amphibian susceptibility to diseases. Chytra is not one that I know has been studied, but a number of studies are showing that pesticide and chemical exposure lower immune function, and then frogs are more likely to get bacterial or viral infections.
Anything else, Spencer?
No, I guess that was it. I was just curious because of BD being an organism itself, if there are any known interesting interactions between that, compared to maybe some other infections that aren't at an organismal level or anything like that. So yeah, that's helpful. Thank you.
Okay, great. Thanks. Okay, so we've got two questions came in by chat. One is, how likely is atrazine to contaminate groundwater? And I know that it's very likely because atrazine is the top pesticide detected in U.S. groundwater. So whatever we're using 80 million pounds or so of atrazine every year. It's impossible to input 80 million pounds of something into the environment and not have it show up somewhere. So that's where it shows up: groundwater, tap water, rainwater, and gravity eventually brings everything down to water, and that's where frogs live. The frogs have permeable skin, so that's why these pesticides can have such a damaging effect on the amphibians. And we'll do one final question for Dr. Hayes. Bees are in the news a lot, and I know recently a pesticide or a group of pesticides was banned, I think, throughout the European Union, or at least temporarily prohibited until their effects on bees are determined. Bees have been dying off. So the question is, can atrazine have a role in bee die-offs? Has there been any work related to atrazine in bees?
I'll get to that. Let me comment on the previous question. Even not only is frog skin permeable to atrazine, but the large levels of atrazine contamination we see in humans and field workers are also because of inhalation and absorption across the skin. So even humans are susceptible to atrazine in the rainwater, or in Australia, for example, where atrazine and a similar chemical are used in swimming pools. So the children are swimming, literally swimming in atrazine, and it can be absorbed across the skin. I don't know of any studies that have looked at atrazine in bees. The compounds that have been associated with the bee decline are so-called nicotinoid insecticides, but I don't know of any studies of atrazine that have looked at bees or impacts on insects.
Okay, can you just clarify that, what you just said about pools in Australia? I know they use atrazine on sugarcane in Australia. I've never heard of it used in pools. Why are they using it in pools?
Yeah, so atrazine and a related compound simazine are used in swimming pools in Australia to keep algae from growing in the swimming pools, because atrazine, of course, is an herbicide. So I've testified many times in Tasmania and in southern Australia. It's also used in the tree industry, the forestry in Australia. But the biggest probably impact on children in Australia is the fact that they're swimming in it because it's applied to swimming pools. That's not the case in the United States that I know of.
Okay. So that's our final question for you. I'm going to continue briefly to show people what kind of resources we have for atrazine on our webpage. You're welcome to stay on before you go, or stay, though. If you have any final words for us, then go for it.
No, I want to thank you for the opportunity. I want to thank everyone for being out there. I can be contacted at tyrone@berkeley.edu. So tyrone@berkeley.edu if you want to be in touch. And I thank you for the opportunity again, and I thank everybody for joining us, and I urge you to go out and make a difference. Thank you very much.
Thank you very much. Okay, everybody, that was Dr. Tyrone Hayes of UC Berkeley, tyrone@berkeley.edu. Check out his website, which I believe is atrazinelovers.com. And so we're going to continue. So what you're looking at right now is the Save the Frogs homepage, savethefrogs.com. So what you can do is go to the top of savethefrogs.com, and right there at the very top is a link to submit your comment to the EPA. It's super easy. You go straight to the page, enter in your comment, and click through, and that's just about it. So that's easy. And then how do you find more info on atrazine? Just go to our webpage, and then either in the left sidebar of any of our pages, there's an atrazine link, or from the homepage, just scroll down until you see Save the Frogs, ban atrazine, click through. You'll get to our atrazine page, and you can find a whole lot of info here, general info on atrazine. You can find the link to our electronic petition. So if you've never signed that, please click through, sign it, share it by email, share it on your social networks, send it out as an email to your friends and colleagues, students, professors, ask them to sign the petition to ban atrazine. There's a link to a PDF of the slideshow that I used when I spoke at the EPA last summer. There's transcript of text from my speech. There's a downloadable MP3 of my speech at the US EPA, and there's a link to the YouTube video. It's a 25-minute presentation, which I consider as one of the best presentations I ever gave. It was certainly one of my favorite. So I strongly urge you to go listen to it. And there's some comments that I submitted to the EPA in 2011. You can download those. You can download a letter from Congressman Keith Ellison of Minnesota that he sent to former EPA Administrator Lisa Jackson, urging her to ban atrazine. We even have a link to a sample comment if you want to go grab some text from that comment so that you can comment to the EPA, which, to remind you, your comments due August 26th. You can go grab that. There's a printable PDF of our atrazine petition, so you can print that up or just pass it around to some friends and ask them to sign it. Take it to a live event of yours and get signatures. We've got an atrazine flyer that you can print out 8.5 by 11 inches, post it up around town to help spread the word. We've got really awesome info cards, 4 by 6 inch info cards that have some of the basic facts about atrazine. They are eye-catching. You can order them right through this link. They're $10 or so for 100 of them. It's a pretty good deal, 10 cents each. That covers our cost. If anyone wants to organize a Save the Frogs Day rally, that would be fantastic. And the time when I would suggest for you to do it is Save the Frogs Day, April 26, 2014, or the International Day of Pesticide Action, October 12th. So if you want to organize a rally, then read through all this page, think about what you'd want to do, and then send us a line. Yeah, we also have a bunch of studies linked to on this page. If you scroll down, there's a summary of atrazine from Dr. Rohr at the University of South Florida that I think is well worth reading over. And we've got some presentations from Dr. Tyrone Hayes, some videos, a map of atrazine usage, and going back to what Dr. Hayes was talking about with regards to environmental justice, I think this is very interesting. This red area is the heaviest atrazine usage in the US, and you can see that it's flowing straight down the Mississippi, and those are some of the poorest communities in the United States, and that's where atrazine is ending up. So we've got lots of downloadable PDFs on this page too, and links to relevant articles that you'll find interesting. So on this page, there's a whole lot of information. So please spend, 15 minutes, an hour, 2 hours, however much time you have going through this, thinking about atrazine and what you want to say, and then just go to our homepage, click through the link at the top to submit a comment to the EPA, and do that as soon as possible. August 26th is the last day. I don't suggest waiting until the end. And one final thing that I want to do is talk to you about some upcoming events at Save the Frogs. We now have dates for our 2014 Belize Eco Tour. It will start on June 18th of 2014, and it's 10 amazing days where you will see all kinds of Belizean tropical wildlife and go to some really beautiful places. The Save the Frogs art contest closes on October 15th. So if you're an artist, please submit some cool Save the Frogs art. If you know art teachers or any teachers, please tell them about the art contest and the Save the Frogs poetry contest, which also closes October 15th. They are great ways to get students thinking about frogs. And the 6th annual Save the Frogs Day, April 26, 2014. We encourage all of you to hold Save the Frogs Day events. Save the Frogs Academy is what you are listening to and attending right now. We are very pleased to be offering classes every Wednesday and Sunday all about amphibian conservation, taking place online. They're free. They're accessible to anybody anywhere in the world with internet. So we encourage people all around the world to join us. And just to remind you, for each class, if you plan to attend the class, go to savethefrogs.com/classes or savethefrogs.com/academy and be sure to register for that class. You have to register for each class individually. You're not required to sign up for an entire semester. You can just attend classes whenever you have free time. And all the class videos we will be archiving on our Save the Frogs YouTube channel, youtube.com/savethefrogs. So you can go there and watch past classes. And if you miss a class, all you have to do is go to our YouTube channel to watch the class, including this one, which we will get up to YouTube. Upcoming classes. This Wednesday, learn how to fundraise. And then Sunday, August 25th, learn all about the amphibian pet trade, ethical issues, ecological issues, ways that you can help out. August 28th, California's amphibians, natural history and threats. September 1st, how university students can help save the frogs. September 4th, I will give an introduction to amphibian conservation in Spanish. And about a month after that, we will probably have another Save the Frogs Academy session all in Spanish. And that's something we hope to get going once every month, is a Spanish Save the Frogs Academy class, because we have many supporters throughout Latin America, and we want to make it as easy as possible for them to get involved. September 8th, I will be talking about Save the Frogs Ghana. We will have Save the Frogs Ghana Executive Director Gilbert Adam on the line talking about his work, and also Michael Starkey will be talking about his trip to Ghana earlier this year to help save the frogs there. September 11th, I'll be talking about my PhD research, which was on the chytrid fungus, and we will have a very special guest, Susan Jewell of the United States Fish and Wildlife Service, who is in the Department of the Fish and Wildlife Service that deals with Lacey Act issues, which is to say, making it illegal to ship harmful organisms across federal and state lines without proper permitting or reason. So she'll be talking about the Fish and Wildlife Service's efforts with regards to the chytrid fungus. September 15th is all about how to organize a frog event or a rally in your city. September 18th, I'll be talking about volunteering for Save the Frogs. By September 18th, you should have lots of ideas in your head about how to get involved. We've got lots of ideas too, so please join us to talk all about becoming an official Save the Frogs volunteer. September 22nd, we will be talking about frog dissections, ethical issues, ecological issues, and September 25th, a double whammy, Sharp Park wetlands and our efforts to get non-native bullfrogs banned throughout the state of California. Their importation, sale, and purchase banned. So I hope you can join us for those classes and also spread the word to people you know who like frogs. This is a reminder that Save the Frogs has never received a single dollar from the government. We're very dependent on people like you to help us out. So you can make a tax-deductible donation if you work for the government. We have a CFC number for your combined federal campaign. And we also have awesome organic cotton t-shirts on our website. Go to the gift center or go to savethefrogs.com/shirts. Got lots of cool shirts. Educational posters. We've probably got 7 or 8 different posters that are great ways to spread the word. Tote bags made of recycled plastic bottles to help you reduce your need for paper or plastic at the grocery store. And a final thank you to Dr. Tyrone Hayes of UC Berkeley for giving us his atrazine expertise and for all his years of dedication to amphibian conservation and pesticide awareness. So I'm going to call an official end to the class right now if anyone needs to go, but I'm happy to stick around. If somebody has a question, then go ahead, send it in, and I'm happy to answer any more of your questions. So raise your hand if you have a question or send in a chat, and I'll hold out for 20 seconds, 30 seconds to see what happens. All right. On that note, I thank you all very much for attending Save the Frogs Academy. Go check out savethefrogs.com/academy, savethefrogs.com/atrazine. Submit your comment, and we'll be back here Wednesday, 11 a.m. San Francisco time to talk to you more about amphibian conservation. Have a great week.